So, as usual, we'll start with our learning objective. So today we will discuss the structure of the bacterioglycan, and we try to understand the synthesis of the bacterioglycan. This is very important, okay? We'll highlight the difference of the gram-negative and gram-positive bacteria in relevant to the cell wall bacterioglycan synthesis and explain this difference or differences. And we will discuss the differential methods to identify or differentiate between two groups of microbes that are gram-positive and gram-negative. Okay, to recall from last lecture, if you remember, so we said that gram-negative and gram-positive, when it comes to the cell wall, they have a major difference. The difference is bacterioglycan in gram-positive bacteria is thicker compared to gram-negative bacteria. So this is a major. But gram-negative bacteria actually compensate that by having what? An outer membrane. Great. All right, so we want to see how the cell wall is built. And there is too many names, so just hold on, don't be overwhelmed. We try to break it like in small pieces. So the cell wall is built of what? Built of murine layer that's, we call it carbohydrates building blocks. The carbohydrate building blocks is monosaccharide subunits and peptide. If you look on the name bacterioglycan, so it's peptide and glycan. So the glycan is the sugar bar, and the peptide is amino acid, which we will discuss one. So the monosaccharide, okay, is the two. The first one is N-style mimuric acid. We abbreviate it as NAM. So you're going to hear NAM to the end of the lecture, okay? The second one is N-style glucosamine, which abbreviate as NAC, okay? The four amino acids are L-alanine, D-glutamic acid, D-diaminobutyric acid, and D-alanine. All right, so these are the different components. Sugar, two precursors of sugar, NAM and NAC, and four amino acid. Are we clear? All right. We want to see how these subunits, the sugar and the amino acid, arrange. If you look on, okay, so I don't know if I use the mouse because people at home. All right, I'll try. So when I move the mouse, you'll be able to see it, true? Guys, I need to hear your voice. Don't be silent. Okay, so if you look here, the green color represents the NAM, and I try to have the same color on the slide. So NAM and NAC are alternating in a long chain. So you have NAM, NAC, NAM, NAC. Okay.
Can you use the pointer, please?
So you don't see the mouse here moving? Okay, so the problem when I use the pointer, people at home.
No, not the pointer, not the pointer on the top. Right there.
Well, I'm not really, okay. All right, thank you. So you have the NAM and NAC alternating, to form one strand of the backbone of the, sugar precursor. Those NAM and NAC are connected with a chain connected with beta glycoside bond. All right. The NAM subunit is connected to a chain of amino acids, only NAM, okay? The amino acid is connected with something called peptide crosslink. So you have NAM, NAC alternating. They are connected with beta glycosidic acid. The NAM is connected to the chain of amino acid, and the amino acid between two NAM subunits is connected with what we call peptide bridge. Tamam? All right, moving one step. Okay, one thing that I would like here to take a note of, the peptide bridge connecting two NAM subunits is only available in gram-positive bacteria. It's not in gram-negative. So if you just have this comparison, so this is an E. coli. E. coli is a gram-negative bacteria. You will hear about it when we discuss gram-negative bacteria. Staph aureus, staphylococcus, is gram-positive bacteria. You have the alternated sugar, you have the amino acid or amino acids are connected to the LAM subunits, but there is no cross link. When you come to Staph aureus, which is a gram-positive bacteria, you have the alternating sugar, the amino acids chain, which cross-link by the peptide bridge. Are we clear? Okay, we come to the difficult part. So how is the cell wall, built to have the peptidoglycan? It's a peptide and sugar connected. Now we just talk about these different individual, components of the cell wall. There is 3 stages that, bring us to have, a mesh of the cell wall to protect the bacteria. So those 3 stages happening in different locations. And there is a lot of enzymatic reaction and catalyzations actually happening, help us to achieve the formation of the cell wall. The first stage, which happens in the cytoplasm, and at that stage, the only thing will happen is to formations of the sugar or monosaccharides, subunit. Okay. The second stage would happen in the membrane, and at this stage, the NAM-NAC subunit, which formulated on the cytoplasmic or the stall, will be actually incorporated into a lipid carrier to be flipped to the outside. In the last stage, where the extracellular and extracellular in gram-negative bacteria will be what? You remember we said that gram-negative bacteria have a cell wall and surrounded by outer membrane, but periplasmic space. Thank you. Okay, so let us go and try to see what happened in these different stages. Okay, in stage 1, and this stage we will have what? NAM-NAG, the sugar will be, built or, formulated. We'll have the first thing, we'll have the NAG, okay? So the NAG, which will be formulated from glucose, it's a sugar. The NAM will be attached to UDP to have a component called UDP NAG. So UDP is what is uridine diphosphate, and UTP is uridine triphosphate. So you'll hear these 2 names also. Okay, we have NAM connected to UDP. How that happened? It happened by taking molecules from UDP, and added to the UDP to have UDP NAG, okay? The second step, which will give us the second sugar, is conversions of UDP to UDP NAM. How that happened? By help of enzyme enol pyruvate. The enzyme enol pyruvate will actually catalyze and taking one molecule from enol pyruvate added to UDP NAG to produce UDP NAM. Okay, so at this stage we have the 2 sugar, subunit ready. What next? You remember we discussed, okay, what next? The peptide chain is the amino acid. So the UDP NAM will be attached or conjugated to 3 amino acid. And when this conjugation happens, we'll have what? We'll have UDP NAM tripeptide, because there are 3. Okay? The last stage here, we need to have another 2 amino acids. And those amino acids are built from the conversion of L-aniline to D, alanine. And as a result, when this happens and we have 2 amino acids connected to 3 amino acids, we'll have the UDP NAM pentapeptide. The UDP NAM pentapeptide is a complete subunit of the NAM that will actually have a role in the process. Okay, this is the first stage. So at the end of the stage, we have the 2 sugar glucose are ready, to participate in the process. We move to the, sorry, I forgot something. Cell wall is playing major role in controlling, the pathogenicity. So when you have a cell wall, antibiotic will target process in the cell wall synthesis. So we have 2 antibiotics. The fosfomycin, which used in UTI or urinary tract infection. The fosfomycin will target one of these processes. So fosfomycin will target the step where UDP NAC will be converted to UDP NAM. So this is how the fosfomycin disturb, the second subunit to be formulated. The other antibiotic is cycloserine, and the cycloserine is actually affecting the formation of pentapeptides. So it interrupts the conversion of the 2-alanine to go to 2-D-alanine, and as a result, we will not have the UDP NAM connected to pentapeptides. We'll have only UDP NAM connected to tripeptides. Okay, and we will learn soon why we need these 2, 2 amino acids at the end of the, connected to the tripeptide to have the UDP NAM pentapeptides. Okay, so we're done. We move into the second stage. So second stage happening where? And this in the membrane, and to flip part of the components to the outside. Okay, from last stage, we already have our, sugar, the NAM, of course, NAG also, but we have the NAM connected to pentapeptide, so we have UDP NAM pentapeptide, okay? In order for the complete subunit to be transferred, they need to have a carrier. So in the cytoplasmic membrane, we have something called bacterial. And bacterial is a lipid carrier. The main function of bacterial is to flip the complete, sugar unit to outside. So this conversion or this flipping of the sugar molecule to the outside help with the enzyme called peptidoglycan synthase. Okay, so bacterial will be, attached to the 2 subunits, NAM and NAG, and NAM connected to 5 amino acids called NAM pentapeptide, and this will be flipped or transferred to the outside. So as a result, we have all these, sugars subunits with the amino acid pentapeptide connected, okay? The bacterial, we need to go back. Why? Because the bacteria don't have enough of this bacterial. So they will need to recycle back, and go to their normal location. And in order that to happen, the bacteriophenols will be, recycled by help of enzyme called phosphorylates. Okay? There is two antibiotics again inhibit some of this process to disturb the formation of the cell wall. The first one is the vancomycin, which will actually interfere with the flipping of the sugar to the other side. The other one is bactericin, and bactericin have effect in the recycling of bacteriophenol. Okay? All right. We have everything ready to have a full cell wall. Okay, but still, what do we need? So you have the NAM-NAD alternating. We have these two strands here, you see, and we have the chain of the amino acid. We need the cross-link, and this is what happened in the third stage. Okay, in the last stage, you have everything, the subunit of the NAM-NAD connected to pentapeptides. We need the cross-linking. So the cross-linking actually happens with the help of the transpeptidase enzyme that will, create this cross-linking. The transpeptidase enzyme is also called penicillin binding protein, and there is a lot of penicillin binding protein. So the penicillin binding protein or transpeptidase, so they are the same name, target the beta-lactamase, and you will learn in the pharmacology that there is a lot of antibiotics that work on the beta-lactamase. So this could be where the antibiotic targets to stop the cross-linking of the cell wall formation. All right. Let us just hopefully this will work.
Which one is doing the good thing and which one is responsible?
Okay. This one? Oh, sorry. The bactericin is responsible for the, the recycling, stopping or interrupt the recycling.
What's the deal? Is it made by
I didn't get that. What do you mean?
Is it phosphatase responsible for recycling?
No. Hold on. So in order for the bacteriophenols to come back and recycle, okay, that will happen by enzyme phosphatase, okay? So that will take it back. The bactericin interrupt this process. So the antibiotic interrupt the process of recycling. And if we don't have the recycling, we will not have the possibility of taking these two, sugar to the external to continue the process of the cell wall synthesis. Okay? Are we clear? Yes. I can't hear you. Can you speak loudly? Yes. Oh, okay. So bacteriophenols is a lipid holder. So it's a carrier. So if the two subunits get attached to bacteriophenols, so you have the complete set of bacteriophenols and the subunit will be flipped with the help of the peptidoglycan synapse. Okay? Yes. It's beta-lactamase is one of the antibiotics, targets. So if you have, there is a lot of antibiotics have the beta-lactamase And when you have penicillin binding protein, penicillin binding protein will be targeted to those kind of antibiotics. Yes?
The last stage only happening for gram-positive bacteria.
So that's a good question. When it comes to the how the cell wall, build and the cross link between the amino acid in the gram-negative bacteria and gram-positive bacteria, it's really different from species to species. So if you actually look on, hopefully I have the example here. I can show you like at the end of the lecture slides how gram-positive and gram-negative actually connect the amino acid. The only difference here is what? Is the cross-link between the NAM, okay? Okay, let us just move. Do you have lecture after me?
Yes.
All right, so we talk about how the cell wall is built. We need to know on the significance of the cell wall, or significance of the cell wall glycan, because build of glycan. So it's helping the bacterial growth, as we, just learned. It protects and maintain the cell shape. We discussed that last lecture. Protect the osmotic pressure, so it maintains the ability of the cell to survive in different environments. And also it's provide basic for selection of suitable antibiotics. And this is what we discuss, how different antibiotics actually interfere with the cell wall synthesis. Okay? If I just want to look on quickly on gram-positive, gram-negative bacteria, in the last lecture, we already talked about difference in the cell wall and how the cell wall, build. We just discussed. There is certain actually structure or molecules in the surface of those gram-negative and gram-positive, and we need to know their functions and how they are actually affecting in the pathogenicity of the microbe. So when we talk about gram-negative, we have lipopolysaccharides and something called lipoprotein. Okay, so the LPS, which actually build of three parts: the antigen O, the lipid A, and the core polysaccharides. The most important in LPS is lipid A. Why? Because lipid A is called endotoxin. So when bacteria die and release the lipid A, the lipid A will actually be toxic to neighboring cells. Okay, so the most important in gram-negative, when we talk about lipopolysaccharides, is the components of the lipid A in the lipopolysaccharides, which consider as endotoxin. And I think in the coming lecture, we'll talk about all different, type of the endotoxin and exotoxin. We will discuss in the coming lecture. Okay, so what LPS do, or what endotoxin do? The endotoxin will elicit the innate immune response, and that will be characterized by production of cytokines. Do you know what that means? Okay, so you did that in immunology, true? All right, so when we actually the lipid A and the role of endotoxin win a Nobel Prize in 2011. So that was, a discovery in that time, and the person who discovered that or highlight that in the science was a winner of a Nobel Prize. All right, so what else the endotoxin or the LPS do? It activate the toll-like receptors. And I'm very sure when you actually take your immunology lecture, Dr. Garwin will, talk about the innate immune response and how different, endotoxin or exotoxin could actually activate the immune response, either by production of the inflammatory cytokines or by, activate the receptor to connect to, different antigens. All right. The other thing that I would like to highlight in gram-negative bacteria is grown lipoprotein. And the function of this lipoprotein, it gives the cell more integrity. So it's connected the cell wall with the outer membrane. It's kind of like gives the connections, the tight links between the two layers in the gram-negative bacteria. Okay, moving to the gram-positive bacteria. In the gram-positive bacteria, we have the 6, peptidoglycan, but we have the lipotequic acid or tequic acid. So why they have 2 names depend on the location on the cells and to what they are connected. Tequic acid or lipotequic acid present only in gram-positive bacteria, so it's not in gram-negative bacteria. Okay, it's a polymer of phosphate, ribitol or glycerol, because they are different based on the different bacteria. They covalently attach to the membrane lipid. This is why we call them lipotequic acid. Okay, so this is where the name comes from. And they contribute to the negative charge of the cell external. And we reach when we talk about, the negative charge and the attachment to the positive charge. It's got like, the gram stain. They appear to also have a function in calcium binding. And the most important, they have antigenic, properties. Okay. All right. We talk about cell wall, how the cell wall build, and we discussed in the first lecture and today that the major 2 groups are gram-positive and gram-negative. Gram-positive and gram-negative could be differentiated by using what? By using gram stain. Gram stain considered to be as differential stain. Okay, because it differentiates between what? 2 major groups based on what? Based on the cell wall, structure. Okay. The gram stain actually is not suitable for all types of bacteria, as we also highlight, before, because certain bacteria will have, certain components in the cell wall, maybe like high lipid, they will not be able to take gram stain. And those bacteria will be, stained by special stain, which we will discuss in the coming lecture. The example for that is what? Is the tuberculosis bacteria. They have a very unique cell wall, and they will be, stained by acid path, stain. The gram stain have 4 different reagents, and each of these reagents have function. The crystal violet, the gram iodine, acetone or ethyl alcohol, and safranine. So the end result of gram stain for gram-positive bacteria is, purple color. And for gram-negative bacteria is pinkish, but it's quite difficult, honestly, to be able to see that, pinkish color. So, but the end result for gram-negative bacteria is pinkish. What I meant by this, because if you come to the lab and you kind of like see the pinkish color as red, and you had a question in exam asking you what the outcome of the gram-positive, gram stain for gram-negative bacteria, never say red. It's pink. It's pink. Okay. All right. So if we talk about gram stain, as I mentioned, we have crystal violet. So the first step you will do, you will discuss this in the lab in more details, and you will do it by hand. The first step you will do, you will heat fix your sample. What I mean by heat fixed, you take a slides, a microscopic slides, you take 1 loop. Do you know what loop mean? It's a wire metal, which like long and have like a little circle, and that you use to take, sample for culturing bacteria or preparing like, smear. You will see it on the lab. So you take your sample, you spread it on microscopic slides, and you pass it through flame. This mainly to fix your material on the slides, because you're going to treat it with different reagents, and you don't want your material to, be off. When you actually think of crystal violet is an aqueous solution soluble in water, okay? And it's actually, when they associate in aqueous solution, you will have a crystal violet, positive ion, or positive charge ions, and you have the chloride, which is a negative. So crystal violet, positive will interact with the negatively charged component of the cell bacteria and stain the cell purple. Okay, but I just told you that crystal violet is soluble in water. So when I add crystal violet, if I do the second step by washing, this stain will just be taken off. This is why we add, gram iodine, and gram iodine here work as what? As mordant, and it will actually, the function of gram iodine will increase the binding affinity of the stain. So it will give us what we call crystal violet iodine complexes. Okay? So crystal violet iodine complexes, at this stage, you will have both gram negative and gram positive bacteria stain what? Purple. The step following gram iodine, both gram negative and gram positive bacteria will stain purple. Okay? Following this step, we use a decolorizing agent. And decolorizing agent, as I mentioned, is acetone or ethyl alcohol. Okay? By applying alcohol, the cell wall will be affected. So alcohol will dehydrate the bacterioglycan layer of the gram-positive bacteria. As a result, the cell will be shrinked and trap the crystal violet iodine complex. So you'll have a strong, presence of this violet color or, purple color on gram-positive bacteria. In gram-negative bacteria, the out membrane of the gram-negative bacteria actually will lose or will, have damage on the bacterioglycan. Sorry, the outer membrane will be, lost, and then you will have the tiny layer or thin layer of bacterioglycan. Gram-negative bacteria will become leakage because of the skin. We already lost or affect the outer membrane. So we have the crystal violet iodine complex easy to be leaked outside gram-negative bacteria. And at this stage, as the results of decolorizing agent, we have gram-negative bacteria colorless, while gram-positive bacteria will remain, purple. And the final stage, we will have to have a counter stain, and this counter stain will be taken by gram-negative bacteria. It will not actually affect the purple color of the gram-positive. So when you look under the microscope, or if we go to the first slide, you will see this end result. Gram-positive bacteria will be, purple, and gram-negative bacteria will be pink. Okay, so this is just a summary of the steps and how you do it, the function of each reagent, and you definitely going to have more discussion on this when you come to the, to the lab. All right, why gram stain is important? As we already discussed, that gram stain is initial, characterization and classifications of bacteria, because by using gram stain, I'll be able to differentiate like two large groups, to gram-negative and gram-positive, based on their, cell wall. It's also, help us to identify bacteria under the microscope, because if we use a sample without staining, we will not be able to see details of the, the color and the shape. So the arrangement under the microscope using gram stain, I'll be able to see the different shape of the bacteria, and of course, different arrangement, whether they are single diplococci or cluster staphylococcus, or they are strained like strep. Okay, and the most important, for gram stain is when we screen for infectious agents in a clinical sample, it will be easy to take prepare smear, do the gram stain, and actually identify the bacteria. All right, I think we come to the end, but before I close this session, I got a question from one of Tariq Saleh. Tariq, are you here? Okay, yeah, maybe online. So Tariq was asking, and maybe that's something we need to make sure that we have the right information. Uh, okay, so Tariq message was he was say there were like confusions about the role of or the functions of the cell wall when we state whether it actually allow the movement or the cross of the hydrophobic or hydrophilic. Okay, that's the slide where we talk about outer membrane. And just to clear this, I just would like to say that it's hydrophilic. And this is because of the nature of the cell wall. You remember we said like it's a protein embedded in a lipid layer. So for that reason, so the control is allowing the passage of a very low molecular weight hydrophilic, component through that membrane. Okay. I just would like to ask you, we already complete 3 lectures of bacteria, well, the first lecture was more introduction to the microbial world, where we talk about bacteria and virus and fungi. And for me, virus and fungi was more, and parasitology, of course, like parasites and helminth and protozoa. That was more a kind of introductions. But bacteria part, which will be like, as I said, like the focus. And when we come next lecture, we're still going to talk about different bacteria that will not be stained by gram stain. And we're going to move to talk about metabolism and genetics. And this is considered the basic of the whole bacteriology until we come and discuss so more in details. So, for example, today we talk about lipopolysaccharides and we talk about endotoxin. And endotoxin considered to be like a virus factor. So just try to connect the information. But when we start to talk about the group of gram-negative bacteria, we're going to talk about endotoxin, and there will be like different endotoxin based on the bacteria strain, species, and they will have different effect or different function. So I'll do my best to make the summary as tiny, as clear as possible. But as just Tariq, send me an email. Feel free to email me about any concern or something unclear. Okay, sure.
Doctor, regarding the crystal virus stain over here, the reagent, we use alcohol to leak out the crystal violet.
No, we don't use alcohol to leak out. So as I mentioned, the procedure is built on 4 different reagents, and each reagent has a specific function. So crystal violet itself is a basic dye that used to stain. Okay, but because we know the different structure of the cell wall, we add the other reagents. For example, I mean the staining procedure was built by these 4 reagents. Because crystal violet is an aqueous solution that will be soluble in water, we add iodine to give more affinity of this, okay? Alcohol function is to wash out unbounded stain, but at the same time, it affects, dehydrate, and lead the cell wall of the gram-positive to shrink. But then because gram-negative bacteria will lose the outer membrane and have this tiny thin layer, the crystal violet will be actually moving out or leaked out. So the function of alcohol is differential. So it's decolorizing agent and decolorizing agent. Decolorizing agent means they actually take all unbounded stain out. So if you have actually any type of stain, I mean, crystal violet is just an example, and you will add it to your preparation, you probably will have a lot of stain, are not really, you know, attached together. So you want to wash out this, you know, extra one. So alcohol is decolorizing and dehydrate the cell wall, leading to shrink of gram-positive and losing of the outer membrane gram-negative. More questions? What is the yes?
Beginning, you mentioned a few antibiotics. We should memorize them, right?
Sorry?
In the beginning, you mentioned a few antibiotics.
Okay.
We should memorize them, right?
All right. So the reason I mentioned the antibiotic, because POD is a block which integrates 4 subjects, true? So micro, pharma, pathology, I forget the last, and immunology, sorry. So when you actually, the whole idea of this integration is to connect the information, because when you come to your exam, it will be at the, I think SQ, it will be more of integration. You will not have like a direct, SQ will not be a direct question about micro. It will be micro and, you know, pharmacology, for example, or immunology and pathology. So what I mentioned here is just to give you an idea hearing those antibiotic name. And one, I think Dr. Dalib or Dr. Hatouf come and discuss antibiotic and antibiotic inhibitor. You will recall when she said, well, this, I mean, vancomycin and polymycin and all what I mentioned, they belong to group of antibiotics.